Hello, students. Hope everyone's well today. And I say that on purpose because today we're going to be talking about the lymphatic system. All right, and the lymphatic system is something that helps us to stay well. There we go. Looky there. So we'll be looking at all the aspects of it. There are several functions, but the main one that we want to talk about, the most difficult part of the lymphatic system, will be immunity. Okay, so we are in module 12. I will say that this one is, the anatomy of this one is pretty cool, and then the physiology is fairly complex, okay? So we'll spend a little time on it. It might make this lecture a little longer. Okay, so here's our overview, the big picture, what's going on that we're talking about. All right, the lymph system involves lymph tissue. These are groups of lymphocytes and other specialized cells which support the lymphocytes. So we see where we get the word lymph from, right? Do you remember lymphocytes from our last module when we talked about blood? Remember, lymphocytes are a type of white blood cell, right? And we're going to see how, the white blood cells are very involved, obviously, in immunity. But we're going to see specifically how lymphocytes are involved in the immune system. Okay, the lymph nodes, these are encapsulated masses of lymph tissue found among the lymph vessels. In that picture over here, you can see those little green things, right? This is showing the lymph system and those little nodules right there. Some of those are nodes. The big ones are nodes. And we'll get to some more specifics of that. The lymph itself is a watery liquid formed from interstitial fluid. Okay, so what in the world is interstitial fluid? It just means between the cells. Okay, it's the fluid from between the cells. So where is that fluid coming from? Between the cells. So if you think about it, where do the cells get their water and moisture and this kind of thing? They get it from the blood, right? So the watery liquid formed from the interstitial fluid, that is also coming ultimately from the bloodstream. Okay, so these, we're going to be looking at the differences and the similarities of the circulatory system and the lymphatic system. So take a look. They're working together. They're going to keep us well. Look over here at the circulatory system right here. Okay, we know this. We know this from last time, right? We have our heart. What is the heart doing? It's pumping, right? It's pumping the blood through the vessels and then back to the heart, to the lungs, and back to the heart, to the body. That's why it's circulatory. It makes kind of mostly like a figure 8 kind of a deal. When we get to the respiratory system, we're going to see how that blood, when it gets to the lungs will, of course, oxygenate the blood. All right, so the way the blood gets around the body is through a pump called the heart. Well, the lymphatic system, look how, if we superimpose these upon one another, you could see how these green vessels right here are almost matching the blood vessels, but there's no big pump. What's going on here? We're going to see here how the lymphatic system, the unsung hero of the body, if you ask me, it doesn't have anything to pump it. All right, so we will see how we deal with that. All right, but they are right next to the blood vessels. The blood vessels do affect them in the sense that they can, cause some contraction and things like this. All right, so what is lymph? That's our first question. It's a fluid that comes from the interstitial fluid, and this is the way I like to say it, the serum, which is the liquid portion of blood, right? It leaks out of the blood vessels and bathes the surrounding tissue with nutrients and oxygen. That's the job of the blood, is to deliver all that to the cells, correct? We learned that last time. Okay. Well, what happens is about 90% of that fluid does return back to the blood vessels, but 10% of it or so actually stays in those lymph vessels. And it gets, look at the word, circulated through the lymph system. All right, so why is that 10% of that fluid going through the lymph system? That's our big question. Why is it doing it? And it's because the body is going to test it. It's going to be taking a look at what's happening, ultimately what's happening with your blood, right? Okay, so let's look at our anatomy. Remember, this is the fascinating part. Once again, I have a picture over here of those vessels. Look how they're mimicking, where the vessels are blood vessels are. Maybe not quite as extensive. We have these vessels called capillaries, lymph capillaries. That sounds similar to the bloodstream. They are composed of epithelial tissue overlapping to form a tunnel. Now look at this. See this? That overlapping of these cells causes this little indentation right here that creates a valve. So now picture this. If a fluid is coming through here and it comes through this valve, it can't easily wash back up that way. Do you see that? Because this will kind of flap shut a little bit. Veins work that way too. We talked about that, right? The veins will flap shut so that we can't have any backflow. Well, the similar thing is happening here with these quote-unquote one-way valves. All right, what are our main functions of the lymphatic system altogether? Fluid balance, okay, so keeping the fluid where it needs to be, mainly in the bloodstream. Fat absorption, that's interesting. That would be part of our digestive system, right? And then the big one, the tough one, is going to be immunological defense, or immunology, or your immune system. All right, so let's talk about fluid balance first. Controlling the fluid in the tissue. If the lymph system might become clogged by disease, I'll talk about that a little more in a minute, the end result will be something called edema. Edema. That's the buildup of excess fluid. Extreme cases are known as elephantiasis. Now, my picture's coming in here, y'all. These pictures of elephantiasis are somewhat disturbing, okay? I'm just warning you, all right? So here we go. Here's our first one. Okay, so here's a man or a person that has a problem with his left leg. You see that, right? Okay, so when I say by disease, it almost exclusively, maybe not 100%, but quite a bit of the disease I'm talking about has to do with parasitic infection. Okay, so if there's a parasitic infection, then it can clog up those vessels, those lymph vessels, and then the lymph sort of gets stuck in the tissue, all right? It won't go through the vessel properly. And if it is ignored by apparently this person, it'll build up, and this extreme case is known as elephantiasis. Now, the reason it's called that, you hear the word elephant in there, it's because oftentimes it does affect the limbs, namely the legs, and so the people who suffer from this can have their feet look literally like elephants' legs. Yeah, both feet, sometimes just one, like in this man's case. Our next picture, once again, I have a disturbing picture here. Some cases become beyond extreme. Now, you see this person here, his left hand has a serious problem, right? And you can see it looks like his thumb and first two fingers, 1, 2, 3, 4, 5, have this serious edema. Okay, so edema, of course, has to do with the tissue. Now, it's different than swelling, okay? So we might think, oh, like swelling when I've had, an injury and something swells. No, that's different. That's usually the blood itself coming to the region there. Inflammation, we call that. This is edema. So one of the difference you can tell if you were suffering from this, sometimes down here in Texas, we have really hot summers. And in the hot summertime, we might collect up a little fluid in our extremities, mostly our feet. And some of you may have felt that, maybe your grandma or something like that. If you stick your finger on the tissue and push down and it leaves a little pit, then it kind of slowly refills. That's called pitting edema. And that is part of the lymph system. Generally, with the heat causing it, that kind of thing, we just lift our feet up or we go into the cool room and everything's just fine. Now, these people who are experiencing this, here comes another disturbing picture. Are you ready? All right, so notice his left leg there has a serious problem. So, a couple of things about these last few pictures we looked at. These probably happened in a foreign country because parasitic infections happen there more often, more of a tropical kind of disease, something like that. Possibly in regions that are far away from a good hospital, okay? So these people did not get these things checked out in a timely manner. If they had noticed, the guy with the hand, he had to have noticed that, right? It didn't happen overnight, is my point. And so when we live in a country like America that has modern medicine that's available, we can usually go get those things taken care of. And they can actually do some blood tests. Do you remember last time we talked about blood, which white blood cell tends to show up if we have a parasitic infection? Anybody remember that? That would be our eosinophils, right? If we have a lot of those, there might be some parasites in there. Okay, so now we're back to, remember, the the lymph system does not have a pump, right? So where does it go? It gets pumped, look, it's in quotes there, through the system by way of, ready? Large muscle movement. All right, you can see in this picture over here, the muscle is relaxed and it's contracted and it's going to push that lymph right through that lymph vessel right there. And then it will travel not just through the vessels, but through the nodes. We'll get to the nodes in just a moment. We'll see what happens. A node, they ultimately then will get pumped dumped back into the bloodstream in the largest lymph vessels up near the upper chest, near the clavicles. Okay, so you can see here, I'm going to show you another picture right here where it comes up. The green one is the lymph vessels, and here you can see they'll be dumping in. We have the subclavian trunk, all these kind of things dumped back into the blood system right up in there. Okay, so large muscle movements, here we go. Here's a guy doing jumping jacks. Have you ever noticed when you're sick, you have a bad cold, and you just feel crummy? You know, you just want to just moan and groan and have somebody bring you soup and this kind of thing. But you may have noticed that if you had to get up and move around, you actually ended up feeling better. So here's what I'm going to tell you. When you get sick and you're feeling crummy, just every couple times during the day, do some jumping jacks, okay? Because you know what? That's large muscle movement. That'll get that lymph flowing, and that lymph will flow to the places where it's going to check out what's making you sick and therefore maybe producing the things that will help you feel better. I've often said if you have a trampoline, just go spend 5 minutes jumping on the trampoline. And maybe a jumping jack on the trampoline. That's some good large muscle movement that'll keep that lymph flowing, and that'll allow you to get well more quickly. All right, so here we go. Here is the circulatory, it's simplified obviously, the heart and the arteries and veins and the capillaries. We know how that works. Well, alongside it, we're going to see these lymph vessels with these little nodules and then big nodes. The smaller pieces are just nodules. They're a little bit sort of like dense tissue. The nodes here, it's going to go through that node. Sorry, I've lost my pointer. There it is. Go through the nodes and connect. And look here. It's getting this is where the fluid's coming from. Oh, I was going the wrong way. This is where the fluid's coming from into those vessels, up through the nodes, dump back in up here. Okay? That interstitial fluid is between the cells. It's down here is where that happens. Okay? Fluid exchange between capillaries, lymph, tissues, and lymphs. And this is just another picture showing this area right here. And the nodes are next to it, and it's just going to dump it around, and then part of it's going to go. 10% is going to go into those vessels and nodes of the lymph system. All right, let's look at fat absorption. Let's get the easy stuff out of the way. Special lymph cells in the small intestine called lacteals. They will absorb fat from your diet. It's a milky substance known as chyle, and it eventually gets dumped back into the bloodstream. Okay? Without the system, hard to digest fats would not be absorbed. So it's important in your digestive system to be able to absorb fatty foods, right? We learned from our nutrition aspect of our unit that we talked about that fats are actually quite important for us, aren't they? They don't make us fat. What makes us fat? The carbs and the food that's processed. Now, immunological defense is the big deal, right? It's the big thing we want to look at. It's the process by which the body protects itself, boom, boom, from pathogenic invaders, bacteria, fungi, parasites, foreign substances, in some cases even chemical invaders. All right, so this is going to start to get into the more complicated part of our talk today, okay? So let's look, first of all, if you think about how we get sick, foreign invaders come into our body. Now, how do they get in, all right? Well, into the open areas. Like our skin is completely open to the outside world, right? So our skin, it's protecting us, but if, say, we have a cut or something in our skin, something can get into our skin, right? Well, also, things can get in through our eyes, our nose, our mouths, our ears. Anything that has exit from the body can get infected by external things. All right, so we have this thing called the mucosa-associated lymphoid tissue, or MALT. That's lymphatic tissue. It doesn't have a bunch of connective tissue or capsules or anything like that, but it's in the areas where we have contact with the outer world. So we look at our tonsils are in our throat. Peyer's patches and appendix are in the intestine. That food came in from the outside world into our mouths, down into our gut, right? So we're having that relationship with the outside world right there. Okay, so we have subdivided. This gets a little more complicated than you need to know at this point, but we have these components of MALT. Okay, so if you look at it, MALT stands for mucosa-associated lymphoid tissue. But then we also have here GALT. Who is John Galt? GALT is gut-associated lymphatic tissue. We have BALT, that would be bronchial. That's in the trachea and the lungs and everything. Bronchial-associated lymphatic tissue. Where's NALT? NALT. Nasal, right? Look at all those things I've told you. Those are all things that have contact with the outer world. All right, NALT, nasal, and then we have C, conjunctival associated lymphatic tissue. That's our eyes. They can get infected. Our eyes are pretty good about staying clean because all the tears and that. We're going to talk about tears in just a minute here. We have laryngeal or larynx associated lymphatic tissue. Skin, because it has the outside world, so we have salt. And then we have vault and taut. This is vaginal and testicular, but really it's urinary area because that also has exit to the outer world. Okay, so that's a little bit of a more complex. Some people who study these things might specialize in one of these areas or a multiple of these areas right here. All right, let's look at the tissue. There's several types of MALT, if you will. There's diffuse lymph tissue, unorganized, no distinct boundaries. We have that in our mouth. Lymph nodules have a little more structure. Spherical, like those little green blobs I was showing you in the picture right there. If you stick your tongue inside your bottom lip and move it around, you can feel how it's a little lumpy in there. That's because some lymph nodules are actually in your lower lip there. Peyer's patches in the small intestine, which we mentioned earlier. Our lymph nodes, though, these are a little more concentrated. I think I'll have a picture of this to show you in just a little bit. They're encapsulated by dense connective tissue. If you do have the slide set that goes with this course and you have a microscope, you can look at a good picture of a lymph node. I mean, a good slide of a lymph node right there. It's actually quite complex in its formation in there. It's concentrated lymph tissue. It's encapsulated by dense connective tissue. I'll show you pictures. All right, there's a lymph node to the right there, okay? They have this structure on the inside, a bunch of stuff going on in there. If you feel your neck, you know, if anybody's ever told you, ooh, it looks like you have swollen glands. Well, they're not glands. They're lymph nodes. They're swollen nodes. They should be more accurate in saying that way. All right, so the structure, we have internal structure. We have these things called trabeculae, like Alex trabeculae in here, right? They're forming these little walls, if you will, just structure, almost like a net kind of a thing. Contains germinal centers. Germinal means something's being made there, right? Something's being transformed there, germinal center. This is where lymphocytes can go. So where are lymphocytes made? Where are all blood cells made? In our bones, right? Well, lymphocytes are special in that they can come to a lymph node when needed, and there will be this rapid mitosis and this proliferation of lymphocytes in these centers. And all of a sudden, boom, we can just be pumping out a whole bunch of new lymphocytes to go do their work to take care of anything that's infecting you. So if you've ever had an infection before, you might have had a high white count. Your white blood cell count would have been high. And then they will do what they call a differential or a diff count on you, and they might see a whole bunch of lymphocytes. And if they see a whole bunch of lymphocytes, they know you have an infection going on somewhere. You probably have a fever and that kind of thing. All right, so they can go to the bloodstream now and prepare and take on whatever is bothering you. It's battle time, right? Here's just a closer picture. If you want to pause the video right now and draw that. If your instructor, your parents want you to have a drawing of this, or like I said, if you do have the slides, you can see something. It won't look quite as, it's just this picture is a better way to look at it, because when we try to look at it under the microscope, we have to have a really thin cutting, and it's harder to see all the parts. All right, I think I'm going to talk a little bit more about that. Okay, I think I want you to notice something right here. Notice how we have a lot of incoming areas, right? We have these, they're called afferent lymphatic vessels. I mean, stuff's coming into it. So coming in, coming in, coming in, coming in, and only one spot right here of going out. All right, so the next picture, I believe, shows that as well. Coming in, coming in, coming in, coming in, and one spot that goes out. What that does is we have a lot of information coming in, and it has to go through all of this, and then everything has to crowd down here to get out. All right, it's sort of like a log jam, right? Or when you're driving on the highway and you come to a river and you have to cross over a bridge, everything has to funnel down to one little spot. And that's going to be great, because that's going to keep that lymph fluid in that lymph node longer, and therefore more time to check it out and to see if there's some response that the lymph system needs to make. Pretty cool design there. All right, just a review here of our white blood cells. This is a fancified look at it. Our neutrophils, the most common. Eosinophils, remember if we have a parasitic infection. Basophils and monocytes, those are the big eaters. And there's our lymphocyte. It's the smallest one, and it's the one we're most concerned about here in the lymph system. Let's look at a microscope slide. You can see right here's our lymph. I think that's a lymphocyte. It's a little large. There's our segmented neutrophil. Here's our eosinophil. That is probably an eosinophil that's reached its maturity. That's probably a monocyte. It's been a long time since I had to do this for a living here. Let's see. I'm looking for some lymphocytes over here. I see mostly banded neutrophils. See bunches of them. That's normal. But the second most common one would be a lymphocyte. Here we go. Lymphocyte, lymphocyte, lymphocyte. Right there. Okay, doesn't necessarily look to be overly crowded with lymphocytes. If you had a bad infection, you'd have more lymphocytes than anything, even more than the segmented neutrophils. All right. Cytokines, this is interesting. This subject came up a lot during the COVID pandemic deal. Is about a cytokine storm and these kind of things. They're just a group of proteins. The immune system makes them and acts as chemical messengers. They can be large proteins or peptides, glycoproteins. They will regulate the immune responses. Hematopoiesis, remember, is the manufacture of more blood, and therefore more lymphocytes, and therefore those lymphocytes can go to the lymph nodes and proliferate. You see, so it's just another kind of a backup in the system here to continue to make more white blood cells. All right, here's a picture of a man who has swollen glands, quote unquote. Those are actually swollen nodes. Those are his lymph nodes. We have a whole bunch of lymph nodes in our throat area. That makes sense, doesn't it? We have a lot of contact in the outer world through our head, our nose, our mouth, even our ears, not quite so much, but our eyes. These things will go down our throat, and therefore that's a place right there to have good nodes that will take care of our system. They will filter the lymph to scrub out impurities. That's one of their jobs. They will increase lymphocyte production. PRN means as needed. And then we will spot check the lymph for anything big. So they're going to just be checking the lymph fluid for anything that might be a problem. And they actually filter it out. They'll just kind of keep things behind in those nodes, and that's why the nodes get swollen, because they've kept a bunch of stuff in there. So this guy probably needs to be checked out by a doctor, and he might need a antibiotic or something like that, depending on what he has. All right, here we are once again. This is, I was telling you this already, how many afferent vessels coming in, but only one exit, efferent, headed out. And then macrophages, right, the big eaters, the basophils, will engulf invaders and stop infection as well in the lymph nodes, okay? Our tonsils are one of our first places of attack because they're right there in our mouth. You can see here some lymph nodules inside the tonsils. Look at those nodules. Lots of lymph tissue there. So as we're breathing in or eating something, those tonsils right there will be able to handle some of that to get started on checking out to see if anything bad has come into our system. If you did breathe in or eat something there, then they will get swollen. You'll get something known as tonsillitis. In the old days, if you had several bouts of tonsillitis as a child, then you may have had your tonsils removed. Now, I say in the old days because we've decided, since they are part of the lymphatic system, they are very useful for our body. There was a time in our arrogance that we might have said, oh, who needs them, right? So let's get rid of them. We really try to save them now, right? We try not to do a tonsillectomy these days. So we try to, beef up your immune system in other ways. Not that they never do it. You might know somebody who's had their tonsils removed, okay? So they still can do it if it's just, they're just bothering you a lot all the time. Very sore throat, by the way. It causes a very sore throat. Okay, now we're in part B of the One Day Academy model. We're going to talk a little bit more about some lymph tissue, but then we're going to move into the difficult part of immunity, okay? So let's roll. The spleen. Now, our spleen is an organ in our abdominal area. You can see here it's situated a little bit near the stomach and the pancreas, and then the guts would be right behind there, right? So you can see it's kind of purple. They do that on purpose. It's actually quite bright red. It's a large vascular lymph node. Look at that. We're looking at it as a giant lymph node, okay? What does vascular mean? Do you remember? it has a large amount of blood coming to it. It's a lot of blood coming to it. Vascular, that means huge blood supply. It's got white pulp and diffuse lymph tissue and lymph nodules inside of it. It also has red pulp. That's where all those veins and fibers. It's highly vascular coming in there. It's very filled with blood. It filters blood. Notice here, it filters blood. Not lymph fluid, but the blood itself. So let's call it the master, lymph node, right? Because it's going to filter blood. So therefore, it's going to work much more quickly than the lymph nodes will. It will remove old RBCs, red blood cells, via phagocytosis. So that's where they'll go to die, right? Because they have a very short lifespan. You remember red blood cells don't even have nuclei, right? There's no nucleus inside there because they have a very, very short lifespan. Now, notice where this is. So if this is your stomach right here, then your spleen is just a little bit that would be to your left side. And if you think about it, especially if you're a passenger, I guess, in a car, a passenger in a car, and you have the seatbelt on you, and let's say you get into a collision and the seatbelt, catches you. Well, there are times when that catches you so tightly that it can actually damage your spleen. So I had a friend one time who had to have his spleen removed because it had been damaged and he was bleeding internally. And so they actually had to remove his spleen. But he had problems with the immune system after that, right? He had some serious immune issues after that. It's also a oxygen-rich reservoir, okay? So there's so much blood in there, it's going to hold on to that oxygen. For us, it doesn't necessarily, for humans, it doesn't necessarily have an advantage, but there are some creatures out there who have large spleens. For instance, I think it's a seal, like a marine seal, that has a very large spleen, and they hold that oxygen-rich reservoir inside their spleen. They can dive deeply, deeply under the water for a long time and just kind of squeeze their spleen to keep their blood oxygenated so they don't have to come rushing up for a breath of air. Just an interesting little bit of trivia there. All right, here's where you can see where it is, and you can see how maybe your seatbelt coming across you could have a problem there. And here's the internal tissue. We have, once again, we have trabeculae giving it some sort of structure on the inside. We have our red, the pulp, and our white pulp. Okay, so very, very vascular. Oh, here's a normal spleen right there. See seatbelt, boom, like that. This is called splenomegaly. Megaly means it's just very large. So sometimes the spleen will become enlarged for some reason. Typically some sort of disease will cause this, and sometimes people will have to have their spleen removed from that. It's painful and otherwise not good, but hopefully they can figure out what's causing it and therefore make you well and you don't have to lose your spleen. If you can keep your spleen, keep your spleen. All right, our thymus gland, we learned about this in the endocrine unit. Remember, we talked about it being in our chest and how it tends to kind of waste away over time, but it need not if we take the right vitamins, right? It's large in our youth. It shrinks as we age, but we should take our selenium and our zinc and our vitamin D. So immature lymphocytes travel there. So the lymphocytes are made in the bloodstream, right? They may go get proliferated into the lymph node, but they will come to the thymus gland, and there's this mysterious process called a thymic education. It changes them, okay? It makes good lymphocytes mature, and the bad ones are, there are bad ones that are destroyed, okay? So it turns it into a super lymphocyte. All right, the super lymphocyte, that we'll get to the details of it here in a little bit. Just remember, so we would call that now, that the lymphocyte that's had a thymic education is now going to be called a T cell. Don't forget that the thymus gland also has endocrine duties as well. All right, here comes the tough part, folks. So if you want to take a break and finish this next week or whatever, you might want to break right here because we're going to be looking at immunity. The immunity, first of all, things may have changed even since the publication of this book. There's advances happening all the time, and so just keep that in mind. So if some of this that's in our book might be an error, it's because new things have been learned, okay? So you can show some grace on that. All right, we're going to be looking at innate immunity versus acquired immunity. So I have a little Venn diagram over here. We're going to see that we have innate. That means it's just everybody's got it. It's ready to go. It's quick. Adaptive or acquired means that we have to make something a little longer acting. And then in the middle, we have something that's part of both that we'll be looking at. Look at that natural killer T cell. If it's a T cell, where has it been? It's been to the thymus gland. It got its education there, and it turned it into an assassin. Woohoo! Yeah, a good thing for us. All right, innate immunity is non-specific, okay? That means it's going to respond to things in a general sense. It's the same regardless of the pathogen or toxin. It may have, it's designed from birth to protect, all right? Whereas acquired immunity becomes specific. Now that takes a little longer. Notice it's called adaptive up here for some reason, okay? But it's going to take longer. The immune response is targeted to a specific pathogen or a toxin. So in this particular case, it can only do that if it's already been exposed to it. All right, we'll be looking at the details of this complex thing that goes on, and we'll be looking at the idea of vaccines here in just a minute. All right, so innate, non-specific. Let's go back to the easy side, right? So the first line of defense, we have barriers, our skin, sweat, oils from our oil glands, mucus, gastric juices, tears, urine, symbiotic organisms in our gut, for instance. Our second line, we have this fascinating stuff called complement. We'll get a little more detail in just a minute. That's a complex of 20 plasma proteins. They will lyse bacteria, means to explode them. Promote phagocytosis, that means to increase the big eaters, right? The monocytes that will come in and just gobble up a bunch of little tiny bacteria. And they also promote inflammation, which in this case would be good inflammation because it's bringing the blood cells there. It's basically an antibacterial defense. We also have other white blood cells just doing their job of phagocytosis, and we're going to see the specialized part of the lymphocytes here in just a minute, but also a chemical called interferon. When I was in school many moons ago, everybody thought if we could learn how to manufacture or synthesize interferon, we could have a cure for the common cold. And we have actually invented it, but it hasn't worked very well, so we'll see what it does. And then inflammation can be local or systemic, right? So local inflammation would be you got a bee sting and you got a swollen hand from it. Systemic would be fever. All right, let's watch a little video about tears.
Moist and healthy. The lacrimal gland and other small glands inside the eyelid and on the white part of the eye constantly produce tears to keep the eye healthy. Tears drain from the eye through two small openings called upper and lower puncta, located at the inside corner of the upper and lower eyelids near the nose. Tears move through a passage called the canaliculus and into the lacrimal sac. They then drop into a tear duct called the nasolacrimal duct and drain into the back of the nose and throat. That's why your nose runs when you cry.
All right. Did you always wonder that about your runny nose when you have tears? Okay. All right. I mentioned interferon. It's chemical, right? It's this is when we talk about bacterial infection versus a viral infection, there are different pathways by which we're infected, and blood tests can generally tell. Like if you have a high white count, that's usually a bacterial infection. If you have a low white count, it's usually a viral infection. Okay, that's the flu and stuff like that. So, but we'll be looking at interferon. It's an antiviral defense. It's secreted by infected cells. Now, these cells will sacrifice themselves. They die. But they will do something to help nearby cells. And what they will do is, first of all, the interferon can burst bacteria. So that's good. That's an attack right there. They can just put a call out to other phagocytic cells, monocytes, to come to the area, and they cause inflammation. And that inflammation will bring the blood to the area, okay? So that chemical is kind of doing a lot there. So here it is. Here's our infected cell, our poor cell that's going to die. The virus attacks it. The interferon is made, and it sends a signal out. Boom. Put up a barrier. The infected cells need to die, right? Here we go. The recruitment. There it is. Recruitment of white blood cells to stimulate long-lasting immunity. So two different things. One, they can come engulf the thing. Or two, the lymphocyte can come and do what the lymphocytes do, the magic, which we're getting to. I haven't really discussed it because it is very complex. Okay. So there's our interferons. Now this video is a little bit long, but it is very informative. So just relax and watch it.
Every living being needs to fight off other living beings that want to feast on them. So as multicellular life evolved over billions of years, it came up with ways to defend itself. Today, humans have a sophisticated defense network, like physical barriers, defense cells, and weapons factories. But one of the most important defenses of our body is largely unknown: the complement system. It evolved over 700 million years ago and is an army of over 30 different proteins that work together in a complex and elegant dance to stop intruders. All in all, about 15 quintillion of them are saturating every fluid in your body right now. Guided by nothing but chemistry, these proteins are one of the most effective weapons we have against invaders. Many other parts of the immune system are just tools to activate the complement system. But it's also really dangerous. Imagine having trillions of little bombs inside your blood that could go off at any moment. So our cells use numerous mechanisms to prevent complement from accidentally attacking them. Okay, what exactly does it do, and what makes it so dangerous? In a nutshell, the complement system does three things. It cripples enemies, it activates the immune system, and it rips holes in things until they die. But how? After all, these are mindless proteins randomly drifting around without will or direction. Well, this is actually part of the strategy. Complement proteins float around in a sort of passive mode. They do nothing until they get activated and change their shape. In the world of proteins, your shape determines what you can and cannot do, because shape determines what you can interact with and in what way. For example, in your passive shape you might do nothing. In your active shape, however, you might, for example, change the shape of other proteins, activating them so they can activate others. Mechanisms like this one can start cascades that spread very quickly. Imagine the complement proteins as being like millions of matches very close together. Once one catches fire, it ignites the ones around it. They ignite more, and suddenly you have a big fire. To show the actual mechanisms of the complement system is a tad complicated and overwhelming, so we'll simplify here. Now, let's imagine you've cut yourself and a bunch of bacteria enter the wound and make it into the surrounding tissue. Our complement attack begins with C3. C3 is the first match, the initial spark that will start our fire. And to do that, C3 needs to switch from passive to active. How this happens is complex, but let's just say it can happen randomly, through other complement proteins that bind to enemies or through antibodies. All you really need to know is that C3 breaks into two smaller proteins, C3a and C3b, that are now activated. One of these parts, the C3b protein, is like a seeker missile specialized in bacteria, fungi, and viruses. It has a fraction of a second to find a victim, or it will be neutralized by water molecules. If C3b does find a target, it anchors itself very tightly to its surface and doesn't let go. By doing so, the protein changes its shape again. In its new shape, it's now able to grab other proteins and start a small cascade, changing its shape multiple times, adding other complement proteins to itself. Finally, it transforms itself into a recruiting platform known as C3 convertase. This platform is an expert at activating more C3 proteins that start the whole cycle anew. An amplification loop begins. Soon, thousands of proteins cover the bacteria. For the bacteria, this is very bad. It can cripple the bacteria and make them helpless or slow them down. Imagine being covered by thousands of flies. But there's more. Do you remember the other part of C3? The C3a protein. C3a is like a distress beacon. Thousands of them flood away from the site of battle, screaming for attention. Passive immune cells notice the C3a proteins and awaken from their slumber to follow the protein tracks to the site of infection. The more alarm proteins they encounter, the more aggressive they get. This way, complement guides reinforcements exactly to the place where they're needed the most. So far, the complement has slowed down the invaders and called for help. Now, it's beginning to actively help to kill the enemy. The first immune cells to arrive at the battlefield are phagocytes, which means cells that swallow you whole, trap you in a tiny prison, and then kill you with acid. But to swallow an enemy, they need to grab it first, which is not easy because bacteria prefer not to be grabbed and are sort of slippery. But now the complement that has anchored itself to the bacteria acts as a sort of glue that makes it easy for the immune cells to catch their victims. But But it gets even better. Imagine being covered in flies again. Now, imagine them turning into wasps. Another cascade is about to begin. On the surface of the bacteria, the C3 recruitment platform changes its shape again and begins to recruit new proteins. Together, they begin the construction of a bigger structure, a membrane attack complex. Piece by piece, new proteins, shaped like long spears, anchor themselves deep into the bacteria's membranes, until they rip a hole into them that can't be closed again. Fluids rush into the bacteria, and their insides spill out. They bleed to death. The remaining bacteria are maimed and distracted by the complement and quickly taken care of by the arriving immune cells. The invasion has been nipped in the bud before it had the opportunity to become dangerous. You probably didn't even notice it. But while bacteria are not happy about complement, the enemies it might be the most useful against are actually viruses. Viruses have one problem. They need to travel from cell to cell. Outside of cells, they're basically hoping to randomly bump against a cell to infect by pure chance. Here, they're completely defenseless. And here, complement is able to intercept and cripple them so they become harmless and guide the immune system to devour them. Without complement, virus infections would be a lot more deadly. But wait, if we have such an effective weapon, why do we ever get sick? The problem is that in a war, both sides adapt. For example, when the vaccinia virus infects a cell, it forces it to produce a protein that shuts complement activation down. This way, the virus creates safe zones around the cells it infects. When it kills them and tries to infect more, it has a higher chance of being successful. Or some bacteria, for example, can grab certain molecules from the blood that keep the complement system calm and make themselves invisible. So the complement system, while being extremely important, is only one player in the complex and beautiful organization that is our immune system. A beautiful example of how many mindless things can do smart things together.
Wow, okay. Good thing we don't have to know all of those things, right? But we were talking about that other thing called complement. Sorry, oops. Uh-oh, I missed the complement. Maybe it's coming up. That's interesting. I feel like I missed a slide somewhere. Okay. All right. I think we'll get to it. Just keep in mind that complement is a series of 20 plasma proteins formed by the liver. It can control what we call a cascade of biochemical events. And anytime you hear about a cascade of biochemical events, you should groan. Oh no, because there's lots of complexity to it, okay, like we saw in that video right there. What I liked about the video is it discussed things we've already discussed. In other words, when it said that it pops a hole in it, it drains out, that's lysing a bacteria. When it said it was slippery and wasn't going to be caught, that's called the phagocytosis. So that's why I like that video. Also, keep in mind that that's our best guess at this time. We don't know if everything that's said in there is 100% true, because science never is 100% accurate, right? It can't be. It's always changing. It's always subject to change and questioning. All right. So we're still in non-specific immunity at this point, and we're going to be looking at our killer cells. Okay, a natural killer cell, remember, it was formed in the in the blood, it's a lymphocyte that was formed in the blood, and then of course it goes through the thymic education in the thymus. So in this picture, this is a scanning electron micrograph. This, I believe, this is it, the big one, and it's attacking something smaller, but it will attack tumor cells and virus-infected cells. So our natural killer cells are monitoring our body. Anytime any kind of weird cell pops up, like a cancerous type cell, it will go in there and attack it and take care of it. Okay, so we're constantly attacking cancer cells, and sometimes, of course, if they become overwhelming, the cancer cells, then you will be diagnosed actually with cancer. Our eosinophils, remember, they have those big old, parts in there that when they spill out, they can cause, different reactions to take place, including inflammation and stuff like that. And typically we'll see those with parasitic infections. Basophils, they will, look at that, they will also stimulate vasodilation. Vasodilation means your veins will get bigger. And if your veins get bigger, guess what? Your blood can travel more quickly. Higher volume, higher speed. So that's all useful and helpful. Here's our inflammation, the local inflammation of bee stings, and a systemic inflammation would be fever. Fever is caused by a chemical called a pyrogen. It just increases metabolism and the rate of the immune response. I don't know if you've ever noticed, if you have a fever, when you have a fever, You feel kind of crummy, and your heart rate increases. Yeah, it just does because your metabolism is increasing. We see a picture right here of a inflamed hand, and it gets kind of puffy and painful, probably a bee sting or something, and it's red. It's red because the blood supply is going there. Remember, this is good news. We want the blood supply there because it's going to be there to take care of any trouble. The blood delivers the deal. Okay, now we're getting into acquired immunity. This is where things get very complicated. It's also known as humoral immunity. It comes from things we call antibodies. So this thing over here, this shape of a Y right here, is what an antibody is. Our best guess is what an antibody looks like. Okay, so it is a protein. How often do we talk about, important proteins, right? So here's another one of those. All right, so we are looking at 4 polypeptide chains. We have 1, 2, 3, and 4. The variable region is right here. And then the constant region is the darker colors right down here. We have heavy chain and light chain, okay? The variable region is giving it specificity, all right? These antigen binding sites, they are specific to a specific thing that's attacked you. Chickenpox, measles, whatever it happens to be, okay? We have the constant regions have different classes. The constant regions have different classes. IgG, IgM, IgA, IgE, and IgD. Ig stands for immunoglobulin. All right, and these immunoglobulins have, they're in different classes based on how they do their work. How do they, function? All right, so their method of attack, if you will. So the IgD has direct binding. It literally just, like a magnet, just sticks onto it, and usually will destroy the cell. It will cause an explosion, that kind of thing. Or they'll gang up in a giant group, like a giant circle all the way around. They'll glom onto it, and it'll keep it from being able to do anything. It's basically surrounding it. It can activate complement, which is what our video we just watched. I'm pretty sure I have another slide coming up. It can stimulate, IgG stimulates phagocytosis. We have a lot of those big eaters, those giant monocytes coming in to come do that. And then stimulating inflammation. Our IgEs will stimulate inflammation, causing blood supply to increase to a specific area. All right, where do they come from? They're made in specialized lymphocytes, okay? Once again, we have our lymphocytes. All right, here's a giant macrophage right here. We're going to be looking at plasma cells and B cells, okay? Plasma cells and B cells. The B cells, now this is not T cells, right? This is B cells, but still a specialized lymphocyte. They're formed from the stem cells in the marrow. So here's our plasma cell, stem cell in the marrow will form a B cell, okay? And the plasma B cell here, plasma B cell here, release this little black tree-looking thing, branch-looking thing, is a specified antibody. Those Y-shaped things, okay? You can see it kind of looks like a Y, sort of, right? Yeah, okay. For the attack. Now, we also have a type of B cell called a memory B cell. So it won't actually do the work right away. What it will do is it will have a memory, okay? So it will stay in the cell, hold on the antibody for future need. This is why if you have chickenpox once, you won't have it again. And the reason is that your B cells have a memory of it, and they'll start releasing all those antibodies. Okay, so the antibodies are going to be made through our B cell stimulation from the stem cells in the marrow of the blood, and so the memory ones are always ready. And if you should come in contact with chickenpox again, then the B cells will be engaged, and they will start making all those antibodies to fight it off, and you won't get sick. It's pretty cool. In fact, God's vaccine is the best vaccine, okay? The natural immunity is always, always the best way, always. All right, let's watch a little video, shorter.
The humoral immune response is initiated by a macrophage, which takes up an antigen, such as a bacterium or virus, and processes it. The macrophage engulfs the antigen in an endocytic vesicle and partially digests it. Class II MHC proteins bind fragments of the antigen and transport them to the cell surface for display. A cell displaying such foreign antigens on its MHC proteins is called an antigen-presenting cell. A helper T cell can bind the displayed antigen if the T cell has surface T cell receptors that correspond to a specific antigenic determinant on the displayed antigen. Once the helper T cell has docked, the antigen-presenting cell releases interleukin-1, a cytokine that activates the helper T cell. The activated T cell now produces a different cytokine, interleukin-2, as well as cell surface receptors for IL-2. IL-2 triggers helper T cells to divide. The daughter cells respond to interleukin-2 by producing a clone of cells identical to the first T cell in genetic makeup and immunologic specificity. Production of this clone completes the activation phase of the humoral immune response. In the effector phase of the humoral immune response, activated helper T cells help B cells develop into antibody-producing plasma cells. Consider a B cell which has encountered the same antigen as was recognized by the original helper T cell. The B cell processes the antigen, and class II MHC proteins transport the pieces for display on the B cell surface. Thus, the B cell becomes an antigen-presenting cell. If an activated helper T cell has the same specificity as the B cell, it can dock with the B cell and assist it by releasing cytokines. The cytokines trigger the B cell to divide, producing a clone of cells that differentiate into plasma cells. The plasma cells produce identical antibody molecules specific for the antigenic determinant that was displayed by the first B cell, and by the macrophage that activated the first helper T cell. The humoral immune system responds to every antigen in this manner, but different antigenic determinants require different specific helper T cells and B cells.
All right. Wow. Once again, you don't have to know the details of all that stuff, but I want you to see how complex it is. Notice they brought up cytokines, MHC, major histocompatibility complex. They brought up complement. They brought up all those things in there because all of it, they're all working together. Okay, so that's pretty fascinating right there. So one of the best ways to kind of understand this is looking at our blood types. Now, we talked about blood last time. If you know somebody or you can get a hold of a blood typing test, you can just poke your finger and figure out what blood type you are, okay? Because there's type A, type B, type AB, and type O. So there's 4 types of blood, right? And it has to do with what their exterior looks like. Now, we've simplified those pictures up there at the top. Erythrocyte, remember, is a red blood cell. So our blood type refers to the type of surface we might have on our blood cells. Okay, so one of the ways to try to remember these words, like antigen versus antibody, they're very similar, sounding. So I always try to use the word antigen with the word surface. So surface antigen. All right, so we're looking at the antigen when they talked about the bacteria coming in and they were antigenic, right? Well, they had surface antigens. They had something on their surface that our body recognized as foreign. And therefore, since it's a foreign thing coming in, we're going to try to produce something against it. An antibody against it. All right, so let's take a look, for instance, at type A blood. So type A blood has these little markers on there showing it antigen, a surface antigen called antigen A. All right? Antigen A, for this thing right here, will make antibodies. Your body will make antibodies not against itself, but against B. So notice a little triangular shape would fit right into there. Did we watch the video about how, I think we did. In the last module, we watched the thing about how when they gave blood transfusions to people, they couldn't understand why sometimes it saved their lives and sometimes they died. Well, that's because they didn't understand blood types at the time. All right, so here we're looking at if you have type A blood, then you have a marker on all your red blood cells that make antibodies against B. Okay, if you're type B, you have a surface antigen on it that looks, it doesn't look like that. We're just fancifying it so it's a little more easy to understand. But this will make antibodies against A. Now, a good question that we really don't know the answer to is why do they make those and how did they make those? Because even, babies that are born with type A pretty quickly have antibodies to B right off the bat. So we're not exactly sure how that happens. If you're type AB, you have both markers on your surface, both surface antigens, and you don't have any antibodies against other things. So they can receive any blood at all because they don't have any antibodies to attack it. Where conversely, if you're type O, which is a good chance you are, because the majority, when I say the majority, since we have 4 different options here, nobody's over 50%, but I believe somewhere close to 40% of our population in the world are type O, which is interesting because it's the recessive gene, so I know I'm type O, which is boring, and I'll show you why it's boring in just a minute. But I am making both anti-B and anti-A antibodies. Therefore, if somebody tries to give me blood from a type A person or a type B person or a type AB person, I have all of these attackers in my body that will attack them and glop them up, and I will die. Okay? The only blood I can receive as O is type O. That's it. That's all I can receive. And we're not even talking about the other markers like RH factor, that kind of thing. Okay? We're just talking about the surface antigens regarding AB and O. All right, so therefore, type A needs to receive type A blood or type O. B, if they're getting a blood transfusion, can only receive type B or they can get type O. If you're AB, you can get anything. Now, if you're going to get a blood transfusion, hopefully not, and you're type A, they're going to try to find type A blood to give you, okay? Because it's just the best thing to do. Same with B and AB, okay? And O, they have no option. They must give you O. That's all there is to it. That's why it's important to know it. I think our next picture shows, okay, so here's what happens. This is how we test for it, okay? So if you have a little blood typing test, which you can get from a science catalog, and I would encourage you to do that, is if you have, if it coagulates up like this, that means you have anti-A, okay? If it coagulates here, you have anti-B, meaning you're type A, and here they both coagulate, and here nothing coagulates. That's why it's boring. So my blood test, whenever I used to do my blood test with my students, I was always hoping my students had something besides O, because O doesn't do anything it doesn't clump up at all. All right, but these other ones do, and they look really cool on a slide. All right, we're going to get into a little bit of controversy here about vaccines, okay? You know, it's not important how I feel or anything about that, but I just will tell you that vaccines are a part of our world, right? How do they work? Well, the idea here is to utilize the design that God put in our bodies to fake our body out that we already had a disease, so therefore we'll make antibodies against it, okay? So that's the idea. I believe the science behind it is absolutely sound. The concept is good. In some cases, you may say that it was advantageous. There are debates about that even. But they use the feature of the attenuated, of the feature of the immunity for memory, that kind of thing. An attenuated virus means you take the actual virus. Remember, a virus is something that probably is bad for us. Let's talk about chickenpox, okay, for a second. When I was growing up, we all got chickenpox because we didn't have a vaccine against it. And what happened was I got to miss school for like 2 weeks. It was awesome, right? I had bumps on my body. It was itchy and uncomfortable, but I would take a little bath, and I got to sit home and watch cartoons and stuff like that. And then I'm free from I won't get it again because my B cells remember it, and I won't get chickenpox again, okay? But in about, I'm going to say, I wonder what year that was. Maybe right at the turn at 2000 or so, maybe just before in the 1990s. They may have come up with a chickenpox vaccine, and I believe it was the attenuated version. And my point being that chickenpox didn't normally kill anybody, right? It was not a deadly disease. When I was growing up, I knew people who had measles, and measles was a little more complicated and complex and, you know, possibly causing more damage, but oftentimes they would survive it as well. So we can take a weakened virus, and sometimes it'll just take all the DNA out of it, so they can't actually do anything, but they do have those little surface things right there. And those little surface antigens will incite or will invoke a antibody production against it, okay? So we get the antibody, will fight the germ. Here it is. So when the actual one, see this one is empty, this one's full, when it comes, our body will fight against it. There's also ones called mimics that are just really close in their surface antigens that can fool and make our bodies, the body will make the B cells, and it will kind of fool them. So the concept of it is good. The science behind it is sound. What's all the controversy? The controversy is that our bodies, the immune system that God gave us, is so fabulous that we really should let it do the job itself. But there are some diseases that are so, but let's talk about like rabies, for instance. There's some new work being done on rabies. I'm not an expert in it at all. That possibly it's actually a toxin that's causing the problem versus a quote-unquote virus. We don't know for sure. So all this new stuff will be interesting to know. What I do know is that when I was a kid, I got a total of, 8 or so different kinds of vaccines, and now it's like 50 or something. It's a whole lot. And just recently, we are now in February of 2026, and just now the health department, the Health and Human Services Department, and the CDC, the Center for Disease Control, have actually changed the vaccine requirements. And it doesn't mean that you can't get all the vaccines. You can get whatever vaccines you want, but you don't have to get them. There are some that had been quote unquote mandated by certain places, public schools, things like that. If you go into nursing school, you're going to be in contact with a whole lot of sick people, and so they generally require nursing students and new nurses to be vaccinated to the hilt, as many vaccines as they can get. But it kind of overloads our immune system a little bit, and possibly makes our, if I'm always using a crutch, then my body won't get strong again. Do you know what I mean? Does that make sense to you? So in this particular case, as far as I'm not being political in any way, I'm just talking about the immune system and the beautiful design of it. So if you have any questions about vaccines and how you feel about them, or talk to your folks, talk to your parents, see how they feel. It's generally basically a miracle of science, and at the same time, maybe a little bit of an overuse of that scientific miracle. All right, cell-mediated immunity. I know this lecture's going on long. Sorry about that. Cell immunity is conferred by the T cells. Remember, T cells were educated in the thymus, and we have these different kinds. We have active natural immunity. That is, I've been exposed to something naturally. For instance, somebody I know had chickenpox, so I went over and visited with them, and I caught it from them, right? So then my body's going to respond to it. When I was a kid, and when my kids were young, we had chickenpox parties, We'd say, oh, somebody has chickenpox, let's all go over. And we wanted our kids to get it so that they could get over it, and get it while they were young and not when they were older. It's a little harder to deal with when you're older, especially if you have other problems like, asthma or things like that. It can be, the treatment can be a problem. Also, there's active artificial immunity. That's your vaccine right there. We have passive natural immunity, IgG, that's our immunoglobulin G. Antibodies, these happen like during pregnancy, sometimes against a baby, hopefully not. I think we talk about that a little bit with the Rh factor, right, with the bloodstream, yeah. And then passive artificial immunity, antibody therapy, that's some of the new kind of stuff they're doing here, where instead of having a vaccine, we can have some other exposure. All right, cell-mediated immunity, T cells, yay. These are specialized lymphocytes with T cell receptors, TCR, on the surface. All right, we have helper cells. I believe one of the videos talked about that. Our mature B cells and the T cells are considered T helper cells. We already talked about the T killer cells. Notice another word we can use for them, cytotoxic T cells. Yikes. Cytotoxic means it's toxic to the cells, right? So it's a killer. That's our little assassin in our bodies. Destroy virus-infected cells and cancer cells. We have regulatory T cells. They will shut down the immune response, so we don't have over-response of the immune system. Yikes, that's a problem too. Kind of like our allergies can be a problem, right? Our natural killers, right? Natural killers, they pull in the innate immunity side of it. These are also T cells. They're a little bit different from the T cell, the cytotoxic T cells. And then we have our delayed hypersensitivity T cells. That's, if we have an overreaction, is our allergy situation. All right, causing itchy eyes and mast cell production and things like that. All right, major histocompatibility complex. Interesting. I thought we had talked about this already. That was mentioned that in the video at one point. About 20 specific surface glycoproteins on every one of our cells. There is so much happening on our cells. Isn't that crazy? It's more unique than a fingerprint. And it ensures that we don't make antibodies against ourselves. Okay, if we make antibodies against ourselves, then we call that an autoimmune disorder, and it is a problem, okay? And we've been seeing more of that lately. I'll get to it in just a minute. Here's our T cells. Remember, thymic education. Their specialty is distinguishing between the host cell, that's me, and foreign cells, that's the thing that came in, by judging what we call aberrant MHC, major histocompatibility complex, by infected cells. So they will promote T cell and B cell proliferation. That's more of them being made, all right? So here's our B cell, our helper T cell. The plasma cell, remember, is kind of like the big one that's going to decide what antibodies are being made at all. So it differentiates from that B cell. We had a slide on that a little bit earlier. All right, types of immunity. Active, natural is exposure. I think we just said this. Yeah, this is actually restating what I did in a couple of slides ago. Active artificial vaccinations. Passive natural immunity. Oh, look here. When we have the breast milk passing to the baby, we have IgGs in that. Immunoglobulin Gs are in there. Immunoglobulin A. IgG through the placenta. Very helpful to the baby. Passive artificial immunity. A volunteer produces antibodies for another. So that would be if you've been exposed to a possible rabid animal, then you'll have these special immunoglobulins. It's a painful shot, by the way. And it's somebody who volunteered. It's somebody else had it, and or they got it from a sick person. Autoimmunity, this is bad. The body begins to attack itself. They lose the major histocompatibility complex recognition. Examples would be lupus, scleroderma, multiple sclerosis. The sclerosis, that word sclero means to harden. So we'll see some of those. So here we are looking at the autoimmunity. Now, also a little bit controversial, that autoimmunity, we've seen like a very large increase in autoimmune reactions in people. All right, we're trying to understand why so much autoimmunity has shown up in the 21st century. Not that we didn't have it before, it's just we have more of it. And so one of the theories, remember this is just a theory, I'm not making a political statement, one of the theories is we're over-vaccinating and causing our body's immune system to not function properly, therefore it almost like it goes into an overreaction thing. Okay, that's oversimplifying it for sure. If you're interested in autoimmunity, you should take a look at it. I'm sure there's plenty of videos and things like that available online. Just keep in mind that, some people opinion it matters more than research, so just look for the research. Here is lupus. So lupus is an autoimmune disease, so our body's attacking itself, and I thought it generally would affect the skin. It says here a low-grade fever. Photosensitivity means bright lights bother you. You get very tired. Oh, here it is, a butterfly rash on the face. Pleural inflammation means inflammation of the respiratory system. That's not good. Ulcers in the mouth and nose, aches in the muscle, arthritis. So it's not good. It's like this. Notice I've got like an over production of inflammation and these kind of things going on. Yeah, no fun. Let's see. This is scleroderma, I believe, and so we're going to see scleroderma, derma being skin. Maybe this is the one I was thinking of. Sclero meaning hardened. Okay. Who is it? The singer, Celine Dion, stiff person syndrome or something like that. Another autoimmune type situation. So, but for scleroderma, we have calcinosis. That's deposits on the skin of calcium that cause these hardened areas. Raynaud's, the blood vessels in the extremities are smaller, right? So then, look, it's getting they get cold very easily. Raynaud's is pretty common, actually. If you know somebody who gets their fingertips and toes cold, even when it's not that cold outside, yeah. Esophageal dysfunction, acid reflux, right? That's not holding very well there, that little sphincter. Sclerodactyly, thickening and tightening of the skin and fingers. That's kind of stiff person right there, that kind of thing. And however you say this word right here, red marks on the surface of the skin. Telangiectasis. Yeah, I'm not sure. What are we looking at here? This is multiple sclerosis. Yeah, and what happens here, multiple sclerosis. The myelin sheath that's covering the axon of a nerve, right here, it actually gets destroyed, okay? And therefore, remember we talked about this, how the signal travels down the axon, and that these myelin sheaths right here are able to serve as sort of like an insulator on a wire. And it also allows for that quicker response as it jumps over those. You remember all that? Okay. Well, that gets destroyed, and therefore our signals, our electric signals, are not traveling properly. All right. So here's normal. The myelin sheath is covered. Remember, we can have that jump, 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 making it very quick response. But if they're degenerated now, see, the nerve can still have action in it, but it's not being. It's almost like a short circuit, if that makes sense. And it might cause, for instance, somebody who, if you know somebody who has this disease, then you can see, like in their extremities, they might have jerky motions, like some they're moving that's against their voluntary movements. It's involuntary movement that should be voluntary movement. And then multiple sclerosis. Similar, we have these things like this, impairment, fatigue, our eyes. Anyway, none of it's good. Okay, so bottom line is that autoimmune diseases tend to be when our immune system is reacting too much, and it's actually attacking the host. It's not recognizing the host cells, therefore it's attacking the host cells. And this is very bad. Why is it happening? Another big question we're going to figure out. Okay, I believe that is the end of our lecture now. Thank you for sticking through it. It's got some tough stuff. Remember, you don't have to know all of the details, but those videos did cover a lot of the details. All right, very good. I will see you next time.